When insulin binds to its receptors, insulin receptor is activated by autophosphorylation of tyrosine residues, which attract IRS1, that is, insulin receptor substrate 1. This stimulates two signaling pathways. PI3K-AKT, that is phosphoinositide 3 kinase pathway, and the RAS signaling pathway. Firstly, let's discuss the protein kinase pathway. This route is used for short-term effect of insulin, such as increased glucose uptake and stimulation of glycogen synthase activity. The active insulin receptor substrate 1 attracts P85 and PI3. PI3 kinase is activated and phosphorylates PIP2 to form PIP3, that is phosphatidyl inositol 3,4,5 triphosphate. This attracts both AKT and PDK1. PDK1 phosphorylates and activates AKT, that is also known as protein kinase B. Active AKT phosphorylates and inactivates GSK3. Normally, GSK3 causes the inactivation of glycogen synthase that synthesizes glycogen from glucose. When AKT inactivates GSK3, so the level of enzyme glycogen synthase increases. This will lead to more glycogen synthesis and glucose utilization. Insulin generates signals via PI3K, which activates protein phosphatase 1. Protein phosphatase 1 dephosphorylates and activates glycogen synthase. AKT generates signals which trigger translocation of GLUT4 to the plasma membrane, which facilitates glucose uptake. Now, in the second pathway, which is used for the long-term effects of insulin. Ultimately, this increases the glucokinase concentration. Let us see how this happens. Firstly, the IRS1 converts inactive RAS protein into its active GTP-bound form. RAS GTP activates mitogen-activated protein kinase, which then migrates to the nucleus to regulate gene expression. The insulin signal is terminated by endocytosis of the insulin receptor complex in endosomes formed from clathrin-coated pits on the plasma membrane. The insulin is digested, leaving clathrin and the receptor intact. They then recycle to the plasma membrane.